Hello champions, I welcome you all to second PU midterm revision series, and this will be part 3 for biology, where we are going to talk about the final chapter, that is human health and diseases. So here I'll be explaining about all the previous year questions, the most expected questions that could come, and also certain questions I have taken from model question paper. So let's see what's on board. Yeah. Now let's check the paper pattern. So children, over here we have 6 parts. Okay, part A will be having MCQ question. How many? 15 of them. You can attempt all 15 and get 15 marks because you get 1 marks for each question. Then we have fill in the blanks. You have 5 questions. You can attempt 5 questions. And then you have part B, short answer type question, 2 markers. So over there you have 7 options. Out of the 7 options you have to just attempt 5. Please attempt just 5. Don't go without reading the instruction, and don't go attempting all the 7 questions, children. It won't be any of use. So over here 5 into 2, 10 marks you will be getting for this particular thing. Now short answer 3 marker questions are there, 7 questions will be there, out of that just 5. Okay, just 5. So 5 into 3, 15 marks. Now long answer 5 markers are there, 7 questions are given to you, but you have to attempt not 5 but 4 questions, just 4 questions. So 4 into 5, 20 marks. Okay. Lastly, you will be having another 5 marker question. 3 options will be given, in that you have to choose 1. So 1 into 5, 1 into 5, 5 marks. Why have written 15? You, just a second. Okay, works. 1 into 5, 5 marks. So total, how many marks do you get, children? You get 70 marks. Ta-da! If you study. Okay. Now we go ahead with the next thing, that is the weightage of this chapter. So this chapter has a weightage of 11 marks, not something very much small. It is very important. Obviously, when you are studying a very big chapter, you expect lots of questions from this, right? So it is right to expect that, and you will get 11 marks if you study this chapter very properly. It's a very important chapter. Now going ahead, the types of questions that could be framed from this chapter according to the blueprint. So there will be for sure a 5 marker. There will be for sure a 3 marker, and there will be 1 marker question. So 2 marker questions were not there in the blueprints. Okay, so over here you can expect a 5 marker, 3 marker and 1 marker. How many 1 markers? Probably 3 of them. So because 5 plus 3 is 8, 5 plus 3 is 8, and then you, if the total weightage is 11, you need 3 more. So I think it will be coming from over here. Cool, works. But anyways, 1 markers are there for this particular chapter. So what is a common mistake that you do? I want to tell about that and then go ahead. So we think 5 marker is there, 3 marker is there, so something that has that much content, that questions would only come. So you study cancer and you study HIV AIDS and you think like that, only that will come. That's true. Yes, that comes for 5 marker and 3 markers, but the thing is, something else could also come, that is the diseases. The diseases could also come. At this point of time, they would not ask you separate. They will put all the diseases together. They will fill in the blanks. They will ask you, what is the causative agent for this? What is the symptom of this? What causes this? Everything like that. So you have to be very well aware about the diseases as well at the first place. Cool? Does it work? Can we go ahead? Now, no more bug. Let's solve the question. So first question that we have, nowadays diseases like dengue and chikungunya are widespread in different parts of India. To prevent the spread of disease, which vector has to be eliminated? What do you mean by vector? Vector are the one that transfers the disease from one person to another. Okay, now you have 2 diseases. What are disease? One is dengue and the other one is chikungunya. And they are asking you what is a vector that basically spreads this particular disease. So children, the vector for both of them is Aedes. It is what? Aedes mosquito. Now the Aedes mosquito comes in different varieties. One causes dengue, other causes chikungunya. Okay, now you have given different options as well. You have Culex. What does Culex cause? Culex is also a mosquito, and what does it cause, children? It causes. It is not causing, it actually spreads one disease which is known as filariasis, or you call it elephantiasis. Remember the diagram in your NCERT where a person has big, very swollen leg. Okay, that is elephantiasis. Okay, that is spread by Culex mosquito. It is not caused by Culex mosquito. Culex mosquito is having the embryo of basically, the larval stage of the causative agent, which is a roundworm. Okay, it is a roundworm that causes this particular disease. Going ahead, talking about Anopheles mosquito and female Anopheles mosquito, same thing. Basically, they are the ones that causes malaria. Again, it is not the causative agent, it's a vector. It just spreads. It is just coming in between. It is not someone who have to blame. So you do not have to shoot that particular guy. There is someone inside it which is causing all the trouble. Okay. Now you have Aedes. As I said, it's for this. Always remember, children, in the case of vectors, in the case of mosquitoes, the one that feeds on our blood is not the male mosquito, it is always a female mosquito. It is always female mosquito. Male mosquitoes basically feed on the plant saps. So there will be sap inside the plant, no? It feeds on that. Female mosquitoes, since they have to reproduce, they need nutrition. That is why they feed on the blood. Okay, cool. So always remember, if the question was who is the causative agent of malaria, and they are giving you female Anopheles, male Anopheles, just understand this female Anopheles mosquito. Works? So what is the correct answer? This. Going ahead with the next question that we have. A farmer working in a field was bitten by a poisonous snake. Okay, imagine. Now, doctor gave him an anti-venom treatment that contains preformed antibodies. This type of immunization is known as autoimmunity, innate immunity, passive immunity, active immunization. Basically, children, okay, when our body is producing antibodies, at this point of time, you say that this is active immunity. Our body is actively safeguarding us. Okay, so our body is doing the job, okay, our own body. For example, some disease came. Okay, first time I was affected with the coronavirus. At this point of time, my body was releasing antibodies, but it was not very effective, but still it released. So this is active immunity. Next time it comes, my body will remember, okay, this guy has come, I have to kill it. Okay, this is a type of active immunity. Now, the next type of immunity is passive immunity. Passive immunity is where my body is not doing anything. It is passively getting the antibodies and everything from outside. For example, a mother is feeding the child. At this point of time, the child is not having too many antibodies. It's not developed as of now. Okay, so at this point of time, the mother itself is giving her antibodies over there. There are antibodies being transferred. So is the child making it? No. Is its own body involved in this particular process? No. Someone else is giving. The second example is when you do not have too much immunity. At this point of time, okay, at this point of time, you know vaccines. Do you know vaccines? Okay. At this point of time, okay, forget about vaccines. I don't want to talk about vaccines as of now. Okay, next scenario is when the snake bite has taken place. At this point of time also, you do not wait for your body to release antibodies. If you wait for that, you will probably die. At this point of time, I want someone to give me antibodies itself. I cannot wait for my body. So here I am writing, cannot rely on body. Cannot rely on my body. Instead, I am getting things from outside. One is the mother giving the child whatever thing was needed, that is the antibodies. Okay. The next one example is this, where in the case of this thing, anti-venom treatment. What is anti-venom? Basically, it is antibodies plus certain proteins, plus certain other proteins which will be acting against the venom. So it is already made and kept there. I cannot wait for my body to release things. So this is known as passive immunity, and this whole process is known as passive immunization. What do you call this? Passive immunization, children. Okay, so that is one thing. Now, innate immunity, what do you mean by that? Innate immunity is basically when your body from the very start is doing this particular function. From your body from the very start. For example, from the point when you're born, you do not have antibodies and everything released. At this point of time, who protects your body? Skin, the mucus, the tears, everything, they are protecting your body from lot of things. So that comes under innate immunity. All these things that you see over here, where antibodies and everything is required, you do not say it is innate immunity. That is another type of immunity that we have. It is acquired. We acquire this type of immunity throughout our lifetime. There is acquired immunity where antibodies are being made and then our body reacts to it and stuff like that. Okay, in innate immunity, you're just talking about the skin, the tear glands and everything that is already there within us. They are not specific. They do not know. For example, if one pathogen comes, another pathogen comes, it cannot say that you get inside, you stay outside. It is not like that. It is non-specific. It's for all. It will say no one has the right to get inside my body. Whereas in the case of acquired immunity, it is very specific. I know for one particular antigen what kind of antibody has to be produced. For another antigen, another antibody will be produced. Okay, so this example that is given over here, where the farmer is working in a field and was bitten by a poisonous snake, I cannot wait for my body to release antibodies, okay, but I have to give preformed antibodies to that particular person. This is an example of passive immunization, or this is known as passive immunity, my dear children. Cool? Works? Going ahead. Next, in which of the following condition diseases is there a substantial increase in the activity of mast cells observed in the human body? So mast cells, where are they involved? Do you know? Have you got any idea? So basically what happens, I'll tell you. In our body, whenever there is some allergic problems, at this point of time, our B cells, there are certain cells in our body which are known as B cells, which are responsible for releasing antibodies. So this B cells release a type of antibodies, you call this IgE type of antibody. What do you call it? IgE type of antibody. What does this IgE do? This IgE goes and binds on some other type of cell which you call as mast cells. This IgE goes and binds on some other type of cells which are known as mast cells. So now when the pathogen, whatever is causing allergy, comes and binds with it, okay, what will happen? Basically this mast cell will become active and it will release certain things which are known as histamines, and serotonin. What does this histamine do? Basically this particular histamine will make our blood vessels dilated. Okay, our blood vessels which were like this normal, now it has made it dilated such that all the cells which are needed over here can come over here. So similarly, for example, just think that ambulance wants to go and there is too much traffic. What do people do? Basically they give area, they give side, right, so that the ambulance can travel. Similarly over here, whatever important cells are to be needed at this particular point of time, okay, because there's a response over here, my body is wanting some help, so many cells has to come. For that you have to dilate the blood vessels, okay, and for that you are releasing histamines in your body. But sometimes it becomes too much. There's too much histamine being released. I don't want this response as of now. This is over response, okay, over expression of something. Now at this point of time, I say it is allergy. My body would have reacted normally, but this is overacting. That has been happening. Histamine is being releasing. It causes vasodilation. I have a lot of problem. I have itching, this and that, everything because of the same thing. So over here, the whole thing was to say that the mast cells, whenever they are there, they are responsible for causing allergies. Okay. Now if I wouldn't have said like that, probably you might not have remembered it for a longer run, but now I think you will remember it. So mast cell, you can think like it has big muscles and things like that. It is basically making our body super fine, okay, but sometimes it just overreacts. There are some people who say that, oh, I have big muscles, and they just talk a lot, right? So it's like them, okay. So at this time we didn't needed this much amount of histamine, but this is actually releasing so much amount of histamine which we do not need, which is why we have allergy. Cool? But mast cells are actually good. So this is how actually it works. I just started from here, that is B cells. I said that they are releasing antibodies. So yes, B cells first release plasma cells, and then they also release memory cells. The plasma cells are the one that releases antibodies, okay. The antibodies goes and works on the mast cells release histamines and things like that. So not a very direct procedure, When something comes inside our body, there will be one cell that comes first primarily, that is macrophage. Macrophage will come running, it will take everything inside, it will take the pathogen inside, children, it will kill it and show certain features of it, certain proteins. All the pathogens, they will be having certain proteins on them. Okay, and these proteins will be very foreign to us. So if this particular guy is showing this protein off, okay, other cells will come and see and they will get activated. So here TH cells, that is you call them T helper cells. Anyway, you have to learn all of these things, so I am telling you here itself. T helper cells gets active because they see that over here I have a protein, and this is shown by another type of protein which is known as MHC2. So you don't need to remember that. Someone on the macrophage is showing that here, look, there is a protein which is not belonging to our body. There is some problem. We have to alert everyone else. So basically this protein is shown by this, and TH cells get activated. At this point of time, TH cells basically makes B cells activated. Okay, B cells are different type of cells. So we have two type of lymphocytes. One is T lymphocyte, another one is B lymphocyte. So now the T lymphocytes say, hey, B cells become active, and now B cells, when they become active, they release two type of cells again. One is plasma and the other one is memory cells. Memory cells are just there. They'll remember that something like this happened. The next time the body encounters something like that, it won't wait for the whole reaction to happen. Rather than that, it will release antibodies with the help of memory B cells. Okay. So over here, plasma cells are active. Plasma cells are the one that releases antibodies. Over here, specific antibodies being released, that is IgE, because it's an allergic response. Now IgE goes and binds on the mast cells, and I told you what all happens there. So this is how usually thing works. Okay, so B cells cannot do things randomly. They need the help of these cells as well. Cool. So I hope you got it. Going ahead. Now, the vector for dengue fever is, again this question was asked. Now you tell me, female Aedes mosquito, female Anopheles mosquito, male Aedes mosquito, female Culex mosquito. Tell me the guys. Tell me guys the answer. Sorry, tell me the guys I said. So it is basically female Aedes mosquito that causes dengue. Okay. I feel I am very enthusiastic today. I don't know why. Okay. Now there is assertion reason question over here. First assertion is, virus infected cells produce interferons. Is it true, children? Do you think that virus infected cells produce something? Yes. So for example, this is cell, this is A cell, okay, and this is B cell. Both of them are best friends. Both of them, they are best friends. Okay. Now A cells, if it is affected by some sort of virus, what will happen? Basically A cells releases something which is known as interferons. And now this interferons will slowly go and act on the other cells and say that, hey, B cell, be safe. There is a problem over here. I have been infected by a virus. You could also be infected by a virus. So you have to secrete antiviral proteins. You have to secrete what? Antiviral proteins, such that you could be at least safe. You could be at least safe. So interferons, what are they doing? Basically, they're signaling the other cells to be very active. Now there could be an attack. We have to be active over here. That is the role of interferons. Fine. So virus infected cells produce interferons. It is true. And this is a part of innate immunity. I told you innate immunity is something that is we get from the beginning of our life, right? So to your skin, everything we got it. Is it like, do you think that there is some person who got skin recently? No, right? Everyone got it from the beginning, very beginning. So this is part of innate immunity where nothing is specific. Over here also, interferons, releasing of interferons is also a part of innate immunity. We get it from the beginning. We have it in our body. Okay. Now, interferons can cause inflammation of virus infected cells. No, children. They do not cause. I cannot say it completely false, but it is not partial. It is partially false. Okay. So interferons are used for signaling things, and in your NCERT is nowhere written like that. So we cannot go belong to it. So interferons can cause inflammation of virus infected cells. No. They just are released. They just alert other cells. So A is correct. B, it will be false. So both assertion and reason no. Both assertion and reason no. Assertion is true and the reason is false. You can go with that option. And if I see certain other answers, even though I have checked for, but still if I come across something, since I said it's partially true, partially false, I'll tell you the answer once more later on, if needed. Okay. Going ahead, allergic reactions are countered by the administration of. So allergic reactions, in allergic reaction, who was playing the major role? Who was being released? Children, histamine. Have you heard of this particular tablet, that is cetirizine? You might have heard about this particular tablet, cetirizine. The people who have, for example, okay, when I sweep around or when I do this jadu pocha, things like that, at this point of time, I start to sneeze because I am allergic to dust. Okay, and this happens because my mucus gland, everything becomes so very active. I get clogged, my nose get clogged, and a lot of things happen. This happens because my body is releasing what? Histamine. So at this point of time, people ask me, please have cetirizine. Please have cetirizine. Why do they say like that? Because cetirizine is antihistamine, children. It has antihistaminic properties over here. It will combat the effect of this, and hence my allergic responses will be going down. So at this point of time, I need antihistamine. This is the correct answer. Okay. Now children, tell me, are you liking this lecture or are you getting bored? I hope you're not getting bored, and I hope you are studying well as well. So I'm trying to make it very interesting as much possible, because I know it's late night that you're watching this video. So obviously you might be sleepy as well. So yes, on the 7th question, which disease is the patient suffering from who is showing symptoms such as sustained high fever, okay, stomach pain, constipation, headache, loss of appetite and weakness? So whenever I see this particular, understand the key terms, okay, whenever you want to study the diseases, sustained high fever, that is 39 degrees Celsius to 40 degrees Celsius, comes in one disease, which is known as typhoid. What happens in typhoid? Who causes typhoid, children? It is Salmonella typhi. Please remember, write the first letter of the second term in small letters, okay? So it is Salmonella typhi which causes typhoid, and it basically affects your small intestine and things like that, which is why you get constipation, you get stomach pain and everything. Now if you have stomach pain, you have constipation, you won't be feeling to eat, so loss of appetite will be there. If you're not eating something, you will be having weakness, and after some point of time you will be having headache also. Now if you are very much weak, you're not having food, then obviously you will catch other diseases, you will be having high fever also, okay? Now that is the way how you can remember the symptoms of typhoid. Apart from that, if the typhoid goes out of hand, okay, you might have intestinal perforation. What do you mean by intestinal perforation? You know, we all have walls in the intestine, okay? Suppose this is the intestine. So there will be small holes being made, perforations being made, such that basically what will happen? All the things that could affect you could come outside, okay, or basically whatever food is also there, that will also come outside, and it might kill you also. So intestinal perforation and everything comes later when the situations are going out of hand. Now in your anxiety textbook, there are only 2 diseases for which we have a diagnostic test, okay, only 2 diseases. I'm not counting cancer in there, okay. So there are 2 infectious diseases, one is sexually transmitted, other one is not, which has the test given over here. For typhoid, the test that is given is Vidal test. Please remember it, okay. What is the name of the test? It is Vidal test. Another infectious disease, that is sexually transmitted disease, is HIV AIDS. The test that is there is ELISA, okay. I hope you know that. If you do not know that, it's fine, you can study now also, okay. So in this case, when I'm talking about all this thing, so we have studied everything about typhoid. So typhoid is the one that is caused by Salmonella typhi. What is Salmonella typhi? It's a bacteria. It's a bacteria, my dear children. There's a diagnostic tool to test this. This is Vidal test, okay. And these are the symptoms. If it goes out of hand, you get intestinal perforation and you get killed also. And one more thing, there was a person whose name was Typhoid Mary, okay. What was this person doing? Her actual name was Mary Mallon. So she was a cook at a restaurant, not a restaurant, okay, some regal places, okay, back at this point of time. So she didn't used to wash her hands properly. And how is typhoid spread? Typhoid is spread through feco-oral route, as in the fecus that you defecate, okay, the fecus that you defecate, it mixes off with the water or the food that you're using. Probably we do not boil water sometimes and we just drink it off. So basically this water might be contaminated, or the food that we are having, probably that could be contaminated, okay. So basically at this point of time, this person, after going to washroom, she didn't used to wash her hands, and then she used to make food, which is why other people also had typhoid, and lots of people died because of this thing, which is why her name was kept as Typhoid Mary. Even though this sounds gross, but this is how it was spread, and this makes us educated that we need to wash hands before having food, okay. It's very much important. Going ahead, and we should not be having food from outside places, which is not very hygienic. Question number 8. Assertion is chronic use of alcohol by a person leads to cirrhosis. What do you mean by cirrhosis? Okay, liver cirrhosis, you might have heard of it, right? So liver cirrhosis is when your cells, okay, your cells are getting scarred, or your cells are getting damaged, okay. So chronic use, as in when someone is too much into alcohol, they are drinking too much amount of alcohol, this becomes chronic, and obviously chronic alcohol usage could lead to cirrhosis. This is absolutely true, because what does the liver do? You know, liver's function is to, why does it affect liver? Basically the liver's function is to detoxify things. So for suppose alcohol came in, someone drank alcohol, whatever molecules are there went into a liver, and now the liver, what it does is that it adds certain water soluble proteins around it such that this whole thing can be soluble in water and can move out of our body. But every time it does it, it is making the liver cells tired because you are giving too much work to the liver cells. And now if this detoxification does not happen, what will happen? Obviously, the alcohol will get accumulated inside our body, and something is alcoholic. The whole pH, everything is getting disturbed in our body, right? And we will be killed. So liver has to do this, but liver, when it does this, always it gets scarred, it gets tired, and that's when we say the person has liver cirrhosis. Okay, so this assertion is entirely true. Going ahead with the reason, alcohol addiction at times becomes the cause of mental and financial distress to the entire family of the addicted person. So alcohol addiction, do you think it can lead to mental and financial distress to the entire family? Yes. So people who drink, they are not aware of the ruckus that they're creating. Obviously, the people who are associated with them are getting affected in the whole process. So obviously, they're using money, they are creating a lot of problems in the family. Obviously, this would lead to all this stress. So that is true. But is this particular point explaining this point? Is this point explaining this point? No. They are not saying how alcohol is causing cirrhosis. So do you think that the reason is explaining it? No. So what will you say? Assertion and reason both are true, but the reason is not explaining. So B option, assertion and reason are true, but the reason is not the correct explanation of the assertion. Works? Okay. Next question. A patient is suffering from fever, chills, cough, headache, and bluish fingernails and lips. These symptoms are of the disease. So whenever you see this thing, bluish fingernails and lips. So when do you think that our fingernails and our lips will turn bluish in color? When there is not enough amount of oxygen in our body, or when there is no enough amount of oxygen in our body. And when do you think that can happen? In what disease is that happening? I'll say it happens in pneumonia, children. It happens in pneumonia. What exactly happens in pneumonia? Pneumonia is going to affect our lungs. Pneumonia affects our lungs. So over here, suppose this is a lung, okay, and you have the trachea. The trachea opens into bronchi. The trachea opens into bronchi. The bronchi opens into the bronchi. It opens into bronchioles. Okay, what are they opening into? Bronchioles. And at the end, what do you have? Over here, you have the alveoli. Okay, the alveoli basically are surrounded by blood vessels. And the alveoli basically are surrounded by blood vessels. So if I zoom this in, looks something like this. So over here, you have the alveoli, which is a pouch-like thing, and you have the blood vessels going around it. So what happens is that our blood cells comes, okay, our blood cells comes, usually they are all sad because they have carbon dioxide. So what does it do? When we breathe in, we are taking in oxygen. So what is there inside our alveolus? We have so much amount of oxygen. So basically these blood cells, they say we do not have oxygen, please give us oxygen. So oxygen is gone to the blood cells, and what is given back over here? CO2 is given back over here. So oxygen goes to the blood cells, CO2 is given back to the alveolus, and we breathe it out. That is a usual process. But now you think of a situation where this alveolus, which has to be filled with oxygen, is now filled with something that is known as mucus. It is filled with mucus. Okay, do you think that this exchange can happen of oxygen and everything? No. So aren't we making our cells deficient of oxygen? Yes. So this is the whole principle of pneumonia. So pneumonia causing bacteria. Pneumonia is caused by virus also. Okay, pneumonia is caused by bacteria also. But we are specifically, in your book, NCERT book, they have not mentioned about the viral form of pneumonia and things like that. So we have to be specific about bacterial form. So bacteria, we have two bacteria that causes pneumonia. I won't tell you now because I have a question for that. Okay, I'll tell you later on. You tend to think by yourself what are the two bacteria that causes pneumonia. So this is the whole thing, how it works, which is why we will be having cough, we will be having fever, we will be having chills, we will be having headache, and the bluish fingernails and lips. Whenever you see this term, just understand you're talking about pneumonia, children. Now one thing, common cold is also mentioned over here. Does common cold cause us this? No. One thing that you need to understand is that common cold, yes, it is affecting our airways only. It is affecting our airways, but it never goes down to our lungs. It is not going down to, if it is affecting the lungs, you say you have pneumonia, you have some other disease, but not the common cold. Common cold is something that just stays for 5 to 7 days. It's because of virus. It is because of a group of viruses. You call them rhinoviruses. What do you call them? You call them rhinoviruses. So at this point of time, there won't be no effect on lungs. No effect on lungs, and this is a NEET question as well. So those children who are preparing for NEET, you can get to the answer as well. Okay. Now typhoid, if you see, I told you it's a bacterial disease, and there you do not see all these things. Malaria also, you don't get all these things. Please do not confuse malaria and malaria. Also, you have chills. You have chills that occurs, but you do not have bluish fingernails and things like that. So malaria is a protozoan disease. It's caused by a protozoan. Typhoid is caused by a bacteria. Common cold is caused by virus. Pneumonia again is caused by bacteria, the names about which I asked you to remember as of now. So this is the correct answer. I'll write tick, tick, tick. Works? Going ahead. I hope you're understanding, children. Okay, is the speed fine for you guys? Because I heard that too long video might be, draining you out, which is why I am also not explaining too very much, but I'm explaining what is just needed for you guys to understand, and I hope it works, right? Okay, question number 10. A patient is suffering from the infection of the alveoli of lungs and is showing the symptoms of fever, chills, cough, headache, and bluish colored lips and fingernails. Okay, the patient was diagnosed to be suffering from the infection of what? Epidermophyton, Entamoeba histolytica, Haemophilus influenzae, Salmonella typhi. Can you try the answer? Well, the answer is Haemophilus influenzae. I told you there are two bacteria that causes pneumonia. One is this, okay, one is this, that is Haemophilus influenzae, and the other one is Streptococcus pneumoniae. The other one is what, children? It is Streptococcus pneumonia that causes the disease. So I hope you got. So one is mentioned over here, so you have to put a tick on that. Now Entamoeba histolytica, what does it spread? It causes entamoebiasis, or you call it amoebiasis. Okay, you call it amoebiasis or entamoebiasis. So over here, Salmonella typhi, I told you it causes typhoid. If we talk about epidermophyton, okay, epidermo, so epidermis is there, and on the epidermis someone is fighting. So in the case of ringworm disease, what do you see? There are scars on the on our epidermis, that is our skin. You see ringworm like things. Ringworm, do you think is caused by ringworm? There's a worm involved? No, it forms a structure like a worm. It forms ring, and it looks like worm since it is thoda sa enlarged, which is why you call it ringworm disease. But it's not caused by ringworm or any worm. It is caused by fungus, and one type of fungus is epidermophyton. So this one causes ringworm disease. Okay, so this is a fungal disease. This is a protozoan. This is a bacteria, and this is also a bacteria. Okay, now going ahead, what do we have? 11. ELISA technique is based on the principle of DNA replication, antigen-antibody interaction, pathogen-antigen interaction, antigen-protein interaction. Do you know how does it work? I can explain you. Okay, so listen to me. ELISA is for enzyme-linked immunosorbent assay. Okay, I think you will be studying in other chapters as well this. Okay, but it has been coming over here, I just told you. So enzyme-linked immunosorbent assay is not something that you have to mug up. Understand with me. So what did I do is that there are different types of ELISA. I'm talking about one type. I have different things. There is a one box sort of thing. Over here I have antibodies, specific antibodies, okay. I have specific antibodies being kept over here. I know what this antibody is for. This is for the people who have HIV/AIDS might have this antibodies being released in the body. So I know about it. It's just there. Akash. Yeah. So sorry. Yeah, so there are already the antibodies released from a person's body from HIV. Okay, so we know that this particular antibodies will be only released from people who are having HIV infection. Fine. So this is already there. Fine. This is already there. Now, there is one person. I do not know whether this person has HIV AIDS or not. Okay. So I am taking the blood sample. I am separating the antigens. Now I am putting the antigens over here. So you know the antibodies, they bind on the antigens if they are of the same type. So this antigen comes, this antigen, whatever is there, it comes, it binds. But still I cannot understand whether it has bound or not. It's very microscopic things, I won't understand at all. Okay, so it's just there. So this antigen from this particular person has just come and bind over there. Okay, what do you understand? That this particular person has HIV. Because obviously these antibodies are released on response to the antigens that causes HIV, right? So obviously this person has HIV, but as of now I am not sure and it's not visible to me. So what do? I put another antibody. But this antibody that is there, okay, this antibody which is there, it is linked to an enzyme. It is linked to an enzyme, children. Okay, it's linked to an enzyme, and this antibody is also on response to the HIV AIDS. Okay, so basically this goes and binds on the antigen again. So there's a sandwich that is being made. This is antibody, this is another this is antibody, this is antigen, and this is another antibody that is there. So it forms a sandwich. Now what is the use of this enzyme? Basically, to view things, what do? I add the substrate for the enzyme. So you know enzymes acts on some substrate, right? Enzymes basically they act on some substrate and turns it into something else, right? So over here also the same thing is happening. The enzyme is acting on one particular substrate, which is making the substrate convert it into something else, and which is why I am able to see a color. Now, if this coloration is there, what do you understand by this? That this particular person has HIV AIDS. If this person did not have HIV AIDS, then obviously the antigen which was there, which was being produced from this particular person, would not have come and bound on this antibodies, and they would be free. So no other antibodies can come and attach, and no such reactions would be taking place. So if you see over here, the antigen and antibody reactions are the thing which is one major thing that is used in the ELISA technique. So this will be your correct answer. Yeah, and one more very important thing. Now I hope you understand why it is known as enzyme linked. So you are linking an enzyme with the secondary antibody that is coming and binding on the antigen, right? So which is why it is known as enzyme linked immunosorbent assay. Okay, and I hope something, at least some part of it, you will remember forever. Now going ahead, children, where is the cursor? Yes. Dash antibodies are passed from mother to child. So this is the fill in the blanks type of question where I have just shown you the pattern, okay, because I didn't find the questions like that. So over here you have different antibodies, IgG, IgA, IgM, IgD. Similarly, there will be different options given to you. So there will be 5 questions given, okay, and there will be 5 options also given, which might not be related to each other. And you have 5 statements, you have to fill accordingly in the 5 statements. So over here I put all related, but it won't be like this. Cool. So dash antibodies are passed from mother to child through lactation. So what I told you, different types of antibodies are there. In the case of allergic response, you had IgE. Now in the case of this, that is lactation, in the case of lactation, the antibody which is passed is IgA. It is what? IgA. After doing the next question, I will tell you how to remember it. Okay, so it is IgA which is passed from the lactating mother to the child. Now talking about the next question, that is the 13th question. Okay, so dash antibodies are passed from mother to child through placenta. What is that particular thing? That is passed from mother to child through placenta. So placenta is what basically, children, when the mother, this is the uterus. So over here in this, okay, just a second. Okay, so this is mother's uterus. Over here, you know, a tissue is being formed which is known as placenta, which provides nourishment to the growing embryo or the growing fetus. right? So this particular tissue, some antibodies are being passed from the mother to the child, because the child obviously is not that immunologically capable to release antibodies. I told you it's not an innate feature. You do not get it from the starting of your birth. Okay, you acquire it throughout your lifetime, which is why it is known as acquired immunity. Whenever antibodies are coming, that means you're talking about acquired immunity. Okay, so at this point of time, you are getting these antibodies and you're asked, what are these antibodies that you're getting? Which type of antibodies? It is IgG, children. This type of antibody is IgG. So the first type of antibody that a child gets is IgG type, okay? And the second type will be IgA, which they get from the mother while lactation is happening. So how can you remember it, the different types of antibodies? You can remember this formula, gamed. You like to play games, right? So first antibody that is being given is IgG, then it is IgA, and IgM, IgE, everything is there in our body. Okay, that is something else, and IgE is for allergy. That is also something that you can remember. Cool? Works? Can we go on to the next question? Chalo. This is the 13th question, okay? Okay, I messed up with the questions now. I did the change, I don't know. Okay, just a second. Just give me a second. Okay. So this is the 14th question over here. Draw the structure of an antibody molecule. How can you draw the structure of an antibody molecule? The one that is given in your NCERT looks like this. You have to draw exactly like this. It's very, 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 very easy. It's not a humongous task. Listen. First, make heavy chains. So first thing that you need to understand is that all the antibodies, they have two heavy chains. What are them? They are the two heavy chains. Okay, now apart from the heavy, so heavy chains, if you want, you can make in a double format also, like this. Cool. And now you have light chains as well, so you need to make light chains. Light chains are smaller, so this will be a light chain. So over here you can see, and over here also you can find a light chain. Fine. Now the next thing is that both in the heavy chain and the light chain, the upper region that you see will be different. So this area that you see will be different from this area. Here also, in the case of light chain, this area that you see will be different from this area of the light chain. Similarly, for heavy chain also, this area that you see over here will be different from the rest of the area. This area that you see over here will be different. What do you call this region as? You call this region as variable region. What do you call this region as? Variable region. Okay, both here and here. This area is known as variable region. Now going ahead, this area which does not have any change from here to here, okay, it is known as constant region. So what is this, ma'am? I'm not understanding. So basically I told you there are different types of antibodies, IgG, A, M, E, D. Okay, in all of them the constant region is the same. The constant region that you see is the same, but talking about the variable region, they all have different variable regions, which is why they are unique. IgG will be responding to this antigens, and IgM will be responding to some other antigens. They have their own specificity, and this is because of the variable regions that is over there. Now obviously they cannot just move like that, they cannot just stay like that, they have to be connected to each other. So over here you will be having disulfide bonds. You will be having disulfide. There will be two sulfides that will be coming together. So you will be having multiple disulfide bonds over here and some disulfide bonds over here as well. They are linked to themselves also like this, and they are linked to each other also with the help of this. So I am writing disulfide bonds, my dear children. Okay, now one more thing, this guy also cannot be left alone. Obviously it will feel bad. Okay, I'm not of your team, but you can accommodate me. That's the question. So over here also you will be finding at least one disulfide bond. Here also you'll be finding one disulfide bond. Cool, you have to make it, do not forget. Fine. Now the next thing, ma'am, you made everything. Where does the antigen come and attach? Can you tell me that? So basically what happens is that this area that is a variable region, there will be areas which will be free, which will be having certain notches, okay, And on these notches, okay, what do you call this region as? You call this as paratope. You call this as paratope. Paratope. Okay, on this paratope, who comes and attaches? Basically the antigens. They also have certain things which goes and exactly binds on the paratope. Okay, this region that binds on the paratope is known as epitope. Now if you're not able to see it, so basically think, this is the antigen, this is what? The antigen, the foreign particle which is there. It has certain proteins on it, the regions, you call it epitope. Okay, now this epitope goes and binds on the regions on the variable area. So over here you will be having certain regions like this, onto which this particular antigen comes and binds. What do you call this? This is epitope, and this thing that is there in the case of antibodies, you call it paratope. This is how you have to draw the structure of an antibody molecule, children. Now we'll compare it with NCERT. So over here also you saw the same thing. Over here one thing that you can add up is the here also they also have S and S bonds. There is disulfide, as in 2 sulfide should be there. Okay, so make like that. Cool. Rest everything is all good. Works for you? Yes. So you see these are the bonds, and you can mention the C end over here and A N end over here. So obviously they are made up of amino acid. This is a protein molecule. Okay. What do you mean by antibodies? What do you mean by antibodies? Antibodies are basically proteins. This name of the protein is immunoglobulin protein. Immunoglobulin protein, which is why it is known as IgAGMED, whatever. So immunoglobulin is a protein. So proteins are made up of amino acid. Amino acid will be having a carboxylic end and an amino end. So there will be N end over there and C end over here. Works? Now going ahead, what do we have? The next question. Okay, this was 13th question, this is the 14th question, and this is the 15th question. Okay, a farmer while working on his farm was bitten by a poisonous snake. He was rushed to a nearby hospital where the doctors gave him an injection to save his life. What did the doctor inject and why? I already told you, at this point of time, you cannot wait on your body, you cannot wait for your own body to release antibodies. You need a sudden help. You need a sudden help. So at this point of time, obviously the doctor will be injecting what? It's a snake bite, right? So he will be injecting anti-venom. And anti-venom is made up of antibodies, preformed antibodies. It is already formed, okay? Preformed antibodies and what, children? And certain other proteins. Okay, and why is it given? The second part is, why is it given? Because it will take time. It will take time for the person to produce antibodies. To produce what? Antibodies. Okay, and if it's the first time that the snake has bitten, obviously this will be a primary response. So there are two types of responses. Okay, when for example once a COVID affected me, okay, at this point of time my body was weak, I didn't fight back, but still my body tried to release certain antibodies. So the first response that you always have is known as primary response. The first response that you always have is primary response, which is a weak response. Okay, at this point of time, my body will be making, remember the B cell was making plasma cells that was making antibodies and also memory cells. This time my body will be making memory cells. So the next time something comes in, next time the coronavirus came and attacked me, my body's response was quite good because it was a secondary response. So this will be a stronger response because over here my body knows that what antibodies has to be released. It is well prepared for it. So this is the first time, this is a primary response, and primary response will be always low. So it will take time for the person to produce antibodies. You can write, it's an example of primary response. So primary response and primary response are always weak, children. Okay, even if it is a secondary response, in the case of snake bite, you have different snakes, they have different venom, so you need anti-venom for that. So you should be having the preformed antibodies anyways. Cool. Now next thing, name the kind of immunity provided by this infection. So is your body making the antibodies? No, you're getting it from somewhere. This is passive, this is not active, right? Your body is not actively producing anything. So what do you call this type of immunity as? You call this as passive immunity, children. Call this immunity as passive immunity, where your body is not producing antibodies, you are getting it from someone else. You have to explain like that. Works? Going ahead, what do we have? What is a vaccine? Write the basis on which it acts when administered in the body. So you have to talk about vaccine and how it is administered in the body. So this becomes the 16th question, and I feel it is very much a ruckus that is being created over here. Can you give me a minute? I'll change the questions and I will give it to you. Just give me a second's time, okay? Just a second. Yes, now it's fine. So what is a vaccine, and write the basis on which it acts when administered in the body. So what do you understand by vaccine? Vaccine is a whole different term. So over here I want to hide the slide and I'll explain you a bit. Vaccine, my dear children, you know, when this particular infection came, that is disease came, that is COVID. At this point of time, everyone was asked to take a vaccine. Vaccine is basically for those people also who did not get this particular disease. So usually what happens is that when there is a person, that person gets a disease, at this point of time, the first primary response, as I said, will be low, but this person is producing enough amount of antibodies such that the next time this particular disease comes, the antibodies will be able to fight against this particular disease. Okay, now suppose I cannot wait for everyone to get that disease. Obviously, that sometimes the disease can be grave and you can just get killed in the first attempt only. The first attempt can make you killed. So you cannot wait for the antibodies to be produced every time. You cannot wait for everyone to get that disease. So what do? Basically, you give this same person, okay, some type of thing that will make them understand that this thing is very toxic for them and they have to release antibodies. So what could be that thing? Basically, I can administer them with dead microbes. I can administer with dead microbes. So dead microbes also will be having the same proteins. And these proteins basically, obviously, as I said, this will say that I am foreign, this is a signal like that, right? This is an epitope. Basically, this will make my body release more antibodies next time. So for example, this time I administered. Okay, my body got to know that this is a problem. I have to make antibodies. So body makes antibodies. The body makes antibodies. Now next time, when the live bacteria comes with the same antigens on the top of it, okay, same antigens are nothing but the proteins, okay, on the top of it, basically my body knows that these were the antibodies that was produced in the initial time, and it will produce the same antibodies the next time, and it won't be able to affect me anymore. So this is how things work. So either you put dead or else you put live attenuated. What do you mean by live attenuated? As in they are live but they are attenuated. Their toxic properties are lost. The problem causing properties are lost in them. Okay, they won't be active inside our body. They would not do anything. They will be just having the antigenic properties around it. Okay, so basically they will be just having the antigens or basically the proteins that will be able to excite our body to release antibodies. That's all. Nothing else. They won't be showing that particular feature. That is, they won't be causing the disease. They won't be causing any trouble inside our body. So live attenuated form, or basically the dead microbes, are being put inside our body such that our body becomes very much aware of what antibodies has to be produced in the case the actual infection occurs, my dear children. So that is the whole theory of vaccine. Okay, now going behind over here and looking for the answer which is written over here. Let's see. How can you frame your answer? So this is a 2-marker question. So this is not everything that you have to write. You can make it small. Okay. So vaccines typically contain inactivated or weakened. You're making the pathogen weak. Okay. Weakened form of pathogen or parts of the pathogen like the proteins or antigen. So basically you can put the whole pathogen. Pathogen is something that causes, that is foreign, not very, it's not our part, it's something foreign. So pathogen can be inserted as a whole. You have to weaken them. You do not have to make them strong. You have to weaken them and then put it up. You have to inactivate them and then put it up inside our body, or you take a part of it which can excite an immune response inside our body, which can give an immune response inside our body. So that thing you put, and this will trigger an immune response. So this statement you have to write as it is. There is no way to make it short. Now, next question is, write the basis on which it acts when administered in the body. How does it act? So first thing is antigen presentation. When the vaccine is administered, the immune system identifies the antigen in the vaccine. This antigen can be a part of virus, bacterium, or a weakened version of the pathogen. So you already told it. So you don't have to mention that again. Now, second thing, the body recognizes the antigen as foreign and activates immune cells, particularly B cells and T cells. There are two lymphocytes I told you, B lymphocytes and T lymphocytes. B lymphocytes releases antibodies. T lymphocytes, there are two different types of T lymphocytes. One is the T helper cells, which is making the B cells active. There is another T cells, which is known as cytotoxic C cells. Okay, what are they known as? Cytotoxic T cells, or you call them killer cells. What? Killer T cells. Okay, what do? They basically, if the cell is the problem that is making the big trouble, so this T cells goes act on them, releases certain enzymes such that they're killing the whole cell together. Okay, that is T killer cells or cytotoxic killer cells or cytotoxic T cells, whatever you want to say. Okay, so there are either B lymphocytes which produce antibodies, which do not directly go and kill the cell. It produces antibodies. Okay, the next type of cell is T cells, which can do this, or basically can just help a B cell get activated. That's all. So anyways, if B cell is getting activated, as I said, it produces antibodies, which are proteins that bind to the antigen and help neutralize it, as in kill it, basically, some or the other way. Now, T cells help coordinate the immune response and destroy the infected cells. So, T helper cells, basically, they coordinate everything and they help in killing the whole cells. Okay, that is the next thing. Now, memory formation. The immune system forms memory cells which remember the specific antigen such that next time anything comes of this sort, it will create an immune response. So, over here, you have to write this point and this point, and the answer is khatam, complete. Okay, will you be able to do that? Works. So since I think that I have made you confused regarding B and T cells, I want to explain a bit. Okay, so just spare a bit amount of time with me. So remember, you have two type of cells, B cells, B lymphocytes, and T cells. Both are lymphocytes. Both of them are lymphocytes, and lymphocyte comes under which category, children? Leukocytes. Leukocytes are WBCs. We have different type of WBCs. One such type of WBC is lymphocyte. Okay, in lymphocyte you have two categories. One is B, other one is T. Now, if talking about B cells, what do, children? They produce, when they get activated, they either release plasma cells or they release memory cells. Okay, the plasma cells are the ones that produce antibodies. The memory cells will remember that this thing has come, and next time the response will be really fast. T cells, there are three different types. Okay, over here you have T8 cells, which are helper cells. Okay, then TC cells, which are killer cells. They kill. Natural killer cells, and this is different. Okay, don't get confused. So rather than writing that, I'll just write cytotoxic cells. Okay, and the next one is, again, you also have memory cells. The T1 also has memory cells. Now over here, TH1 is the one that whenever one pathogen comes, that pathogen is shown up with the help of macrophages. What do you call them? Macrophages. They are big joined cells which eats up everything and shows them on the body. Okay, T helper cells will be the one which will be able to recognize it, and it will say, hey, B cell, you have to do your work, please release plasma cells and everything. So this type of TH cell works like this. Okay, it gives information, coordinates everything. But when you talk about this cell, okay, in the case of this cell is kind of very angry cell. Okay, what it does is that, for example, this cell is affected cell, this is sick cell. Okay, the TC cell directly goes, okay, releases certain enzymes which are known as perforins. It perforates the wall of this particular cell and kills it overall. So that is how it works. So this is a direct killing. This is a sequenced way of doing things. Okay. So you can write it in whatever format you want to remember. Just remember, the vaccines basically makes your body have an immune response. In this immune response, the B cell and the T cells get activated. Once they are activated, obviously they will be making memory cells which would remind the body the next time if something of this comes, it can give a stronger immune response. As simple as that. Works? Okay, the next question that we have over here is question number 17. Name of the drugs, its source and the nature is given in the table below. Find A, B and C. So name of the drugs given here. So this is from drugs and this is something that we have to remember. I know it's difficult to remember. I'll try to make it a bit easy, okay? So the first type of drugs that we have is opioids. Okay? First type of drugs that we have is opioids, and this is what you get from papaver somniferum. Remember, the S has to be small. I made it big, so let me write it in small letters. It is made from papaver somniferum, my dear children. Okay, and this is depressant as it is written over here. Can you give me an example of opioid? Have you heard of heroin? Have you heard of heroin? Have you heard of morphine? All of them, they are example of opioids. And how do you get opioids? Basically, the papaver somniferum plant, no, it has latex, it has a milky latex. With the help of the milky latex, you do some treatment to the milky latex, okay, you do acetylation and everything. That's when the latex is used, that's when you get these things. Okay, so modified version of morphine is known as heroin, and it is acting as a depressant. You might have heard people who are there in hospitals and all, doctors, sometimes they give morphine to people who have underwent surgery because it is acting as a painkiller. They won't be able to feel that. Okay, they won't be able to feel that pain because of this thing. But obviously, you cannot give to random people like that. Small cuts and all, it does not work. Okay, I'm talking about for big people, because obviously it will have some very big effect on them. They won't be able to regain that consciousness for some time. Okay, so that is there. So opioid is a type of drug and it is a depressant, children. You can remember it with morphine. So morphine makes you all gloomy and sad because you don't want to feel the pain as well at this point of time, right? Works? Okay, and now I wanted to tell you one more thing also which I forgot somehow. Anyways, the second, so here what will you fill? Here you will fill opioids. Name of the drug, opioids, or you can fill the name as heroin, whatever you want to do, you can do that. Okay, now the next category is cannabinoids. Oh, one more thing, yeah. Opioids, what are they going to affect? Your central nervous system. The central nervous system is affected, and also your gastrointestinal tract. Central nervous system is affected, that is why you are not able to feel the pain, right? And also the gastrointestinal tract, that is also affected. Now the next thing, next group of drugs is cannabinoids. Cannabinoids, what happens with them? So cannabinoids, basically, it is a source plant is cannabis sativa. What do you call it? Cannabis sativa. Okay, now over here, what does it do? It has effect on the cardiovascular system, and it's not written in your NCERT, but it also causes hallucination. What are the examples? With the examples you might be able to relate and understand much better. Hashish, charas, ganja, marijuana, all of this thing is an example of cannabinoids. How do you get cannabinoids? Basically, this cannabis sativa plant, overall you can use this. Overall you can use this particular plant, the flower, the leaf, everything. When you dry it up, when you take it in raw format, everything will give you different things like this. Okay, I'm not telling you to use it, I'm just telling you this is how it works. And one more very important thing is that basically it, as I said, it also causes hallucination. It's not written in your NCERT. But what do you mean by hallucination? Something that, for example, I am seeing myself flying, which I obviously cannot do. I am a human being, I cannot fly. So this becomes very much dangerous. For example, I have taken weed and I am crossing the road and I am thinking that I am flying, but I am not flying, I am walking actually. And over here truck, I have a bus and everything coming and hitting me, obviously I will die. So this is what the negative effects of weed and everything is. You are hallucinating, you are not in the real world, and if you are not in the real world, obviously it can lead to trouble as well. So that is about cannabis sativa. Each and every part of this plant is used for making the drugs. Okay, papaver somniferum, as I said, latex of the flower pod is used. Okay, there's a flower pod, releases the latex, the milky thing that is being used. Now the next thing is coca alkaloids. coca alkaloids. That is the next category. In this comes the next drug, which is known as cocaine. You might have heard of it, right? And cocaine, you get it from plant, that is Erythroxylum coca. You have to remember the name of the plants. Again, in that format also question will be coming. Now, what does it affect? It affects the central nervous system, children. It affects the central nervous system just like them. Okay, more grave effect is over there. Over here, the central nervous system, what does it do? It makes the body stimulate and release more amount of dopamine. You know, dopamine is a happy hormone. So if it is being released, dopamine signaling, it does not stop, it is being released again and again and again and again. What will happen? Basically, you will be so enthusiastic, so energetic. And obviously, for example, you go to a funeral there, you cannot be happy, and that person goes there, he'll be happy there also, he or she, whoever it is. Okay, so that is the effect of this. And what happens with the regular use? This is the funny aspect that I told you. It's not actually like that. Basically, what is the problem with that is that people who get addicted to this, they would not be able to find happiness in the small things. They would not be able to find peace, because obviously you are addicted on something for releasing the dopamine. So your body, even if it's released little amount of dopamine, you won't be able to feel that thing. That is the thing with addiction, and you will go on, you will crave for more. Once you are in this loop, there's no coming back. So which is why we say not try. So what is the answer for this? Opioids or heroin? Cannabinoids is from cannabis sativa, and erythroxylum coca and everything gives what affects the central nervous system. I hope this is done. Can we go on with the next question? So 3 marks you will get if you know all this thing. Okay, identify A, B, C, D, E and F in the given below, on the table given below. Chalo. Pneumonia is caused by Streptococcus pneumoniae. What are the symptoms, children? I already told you. What are symptoms? You have cold, you have cough, you have cold, you have cough, you have, in the grave symptom, you have bluish nails and lips. Okay, you have fever, you have chills, everything possible. Okay, so it is caused by Streptococcus pneumoniae. Please remember that. And also Haemophilus influenza. There are 2 bacteria that is causing this disease. So if you are asked, give the example of 2 bacterial disease, what all will you give? Pneumonia and typhoid. That is an example that is given in your NCERT as well. So typhoid is caused by Salmonella typhi. Works. And what does it do? High fever, weakness, headache, stomach and pain. So remember, if you are not aware about all the diseases, won't you lose marks over here? It's a 3-marker question. If you forget one thing, won't you lose marks over here? Exactly. So which is why I'm telling you, know each and everything of this particular chapter. Going ahead, what do we have, children? Over here, common cold is caused by rhinoviruses, I told you. And what are the symptoms? Again, what will be the symptom? You will be having cold, you will be having cough, and you will be having upper respiratory tract problems. Upper respiratory tract infections. Okay, not the low ones, not the lungs or nothing like that, just upper ones. Okay, now ringworm disease. I told you ringworm is basically when you have lesions like this on your skin. It's enlarged lesions. Okay, and how do you get it? With the help of a fungi. It's a fungal disease. So this is bacterial, this is viral, okay, and this is fungal disease that we are going to talk of now. So there are 3 fungi which can cause this disease. One is microsporum. So you can remember small spores of fungi. Okay, microsporum. Next is the fighting partners. Okay, one is trichophyton. What do you call it? Trichophyton. Spelling, if you're not able to understand, it is trichophyton. And the other one is epidermis. It is happening on the skin, so it is epidermophyton. Okay, so you have trichophyton and you have epidermophyton, which causes the ringworm disease. Fine. And what will you have? You have dry scaly lesions on the body parts, redness and itching, everything will be there. Going ahead with ascariasis. Ascariasis is caused by a roundworm, that is ascaris. Okay. And what are the symptoms that you get over here? In the case of ascariasis, what are the symptoms? At least few symptoms you have to write, right? So let's see. It is going to affect your intestine and everything. Okay, so there will be internal bleeding. There will be internal bleeding, my children. Okay, there will be inflammation, there will be internal bleeding, there will be constipation that will be happening in escheriasis. Okay, now dash. Over here you have dash disease, which is caused by Entamoeba histolytica. It causes constipation, cramps, stools with mucus and blood clots and everything. Over here again, what can you write? It is amoebiasis. Amoebiasis, which is the disease that is caused by Entamoeba histolytica. Okay, got it? Will you remember a lot of things over here? Okay, apart from internal bleeding and constipation, you can write loss of appetite and things like that also over here in the case of escheriasis. Okay, so you have to remember the names, and if you want, I can make it a bit easy for you. You can take a screenshot. You have bacterial diseases, two. One is typhoid and the other one is pneumonia. Cool. Then you have viral disease. In viral disease, the example that is given in your book is common cold. Okay, dengue. You have this thing, what do you call it? Polio. So polio and dengue is not explained, but it is given in your book. Okay, and polio is something that is eradicated with the help of vaccine. Polio is completely eradicated with the help of vaccine. So that is viral. Okay, bacterial done, viral done. Then comes protozoan. So protozoan that is given is, one is this thing, amoebiasis. Okay, other one is malaria. Both are caused by protozoans. So this is Entamoeba histolytica, and this will be malaria is caused by Plasmodium, not by the mosquito. Mosquito just transfers things. Okay. Now the next thing is roundworm. Roundworm, or you also call it nematode. Okay, so one disease that is escheriasis. Escheriasis, it is very scary. Small worms coming in the intestine, making a junk there. Okay, and the other is Wuchereria. Wuchereria. Okay, that is the name of the disease causing organism. The disease is filariasis. Filariasis. Cool. And then you have a fungal disease also. That is ringworm disease. I hope you got it. Okay, fine. One by one, slowly try to repeat the names and try to learn. It will be easy. I'm telling you, you can trust me on this. It will be easy for you. Going ahead. Short answer type, 3-marker question. Identify A and B in the diagram given below. Explain their roles in the immune system of the body. What do you think A is? So A is basically the swollen parts. Over here they have mentioned something like this, where this comes. So what do you think this is? This is, my dear children, lymph nodes. What do you call it? You call them lymph nodes. And over here, the B part that you see over here, which is near in the sternum, below the sternum over here. What is sternum? So your rib is attached to something in middle, which is known as sternum, the chest bone. So behind it, what do you have? You have something which is known as thymus, which is known as thymus. Both of them have their own functions. Let me tell you one by one. So basically what happens is that you have your blood vessels. Okay, you have your blood vessels, normal blood vessels, right? So this blood vessels or the capillaries, okay, so what happens is that arteries, they open up into arterioles. Okay, I'm making one arteriole over here. Veins, they open up and they form venules. Okay, and in middle you have so many small chutu things, okay, which are very thin, So I'm just making like this. And these thin things that you see over here have pores. So basically the blood that travels through, which has the plasma, which has the WBCs, which has the RBCs, which has the platelets. Okay, all the cells are there in the blood, and also some pathogens is also moving around in our blood. They're all there. So what happens is blood that is going, when they come across this thing that is known as capillary, what happens is that since they have small pores, the blood basically oozes outside. It oozes outside, but not everything comes outside. Who comes outside? Plasma comes outside, and WBC comes outside, and also some small pathogens, they can come outside. Rest of them, RBC, platelets, they go. Okay, they go to the entire body, they circulate, they do all their things. Okay, but these guys, these three guys that you see over here, they are outside. Now where are they? Basically the blood vessels were going where? In between the cells. So you can imagine that there are cells over here, the blood capillaries came over here, and now you have the plasma floating around, the WBCs floating around, the pathogens, they also floating around everywhere in between the cells. So what do you call the space as? You call the space as interstitial space. Interstitial space, as in the space between the cells. And in between the cells you have all this thing going on. Do I need the fluid over here? No, I do not need it over here. There is no, it's a leakage that has taken place. I have to give it back. I have to give it back. So, and obviously one more thing, if it stays over here, it will cause inflammation. This will cause inflammation. So what do? I need another blood vessel. So these are the normal blood vessels that I am having, arteries, veins and everything. But I need another blood vessels which could basically take all this fluid inside and give it back to our blood vasculatory system. So basically give it back to the vein which goes to the heart and pumps and all things that happens regularly. Okay, so there should be one person who takes it and travels. This blood vessel is known as lymphatic blood vessel. This type of vessel is known as lymphatic vessel. And lymphatic vessel, when they go around, okay, they have certain areas which are swollen like this. What are there in the swollen regions? In the swollen regions you will be having soldiers. These soldiers are nothing but WBCs, children. They are WBCs. You have extra WBCs over there. Okay, why do you think they are over there? I'll tell you. So basically I told you the pathogens which are also there. So I'm marking the pathogens in red. I'm marking the pathogens in red. So pathogens which are sneaked out from here, got inside here, they're traveling, they're traveling, they're traveling, they're traveling. They thought that they won't be caught. But WBCs over here, they will catch and kill them. Okay, what about the other things? Plasma, other WBCs will move around and go back to the blood vessel system only. But these guys are trapped over here. So you have such lymph nodes all over your body. You have so many. Over here also, when you go to doctors, sometimes doctors check whether they touch here and see whether your lymph nodes are swollen. If your lymph nodes are swollen, that means you are having an infection, because obviously it is fighting, which is why it gets inflamed, which is why you have an inflammation over here. You get it? So that is the role of lymph nodes, and your lymph nodes have so many WBCs, especially which type of WBC? Lymphocytes. Works? So this is how they work. Okay, explain the role. This is how you have to explain the role. I'll show you. I have made a written format as well. And now thymus. When you talk about thymus, thymus is a lymphoid organ. What do you mean by lymphoid organ? Lymphoid organ is where your blood cells are made. Okay, lymphoid organs are where blood cells are made, or where blood cells are active. So one type of lymphoid organ is primary lymphoid organ. Okay, one type of lymphoid organ is primary lymphoid organ where, oh, I have not made it over here. Okay. So there are two types of lymphoid organs. Primary lymphoid organ is where your different type of WBCs, RBCs, everything are made. Okay. And one such example is bone marrow. Okay, inside our bone there are areas which are known as bone marrow, where all our blood cells are made. Okay. Another example is thymus. So if bone marrow is the place where everything is made, what is the role of thymus? So the problem with this is that bone marrow makes all the cells, okay, all the lymphocytes, WBC, everything what you can imagine. But the cells, some cells like T lymphocytes, okay, some cells like T lymphocytes, they say that we would not mature over here. We will not mature in the bone marrow. We have to go to the thymus and we will mature there only. So this maturation of T lymphocytes occurs in thymus, which is why it is not T. T stands for thymus only. So bone marrow is a place where everything is made and most of the cells are mature also. But T lymphocytes, when they are made, they go to the thymus to get matured. Okay, so thymus also becomes a category of primary lymphoid organ. So at this point of time, they are not meeting the antigens and they are not meeting anyone from outside. They are not active as of now. They have to go to the secondary lymphoid organs to be active. And what is secondary lymphoid organ? Lymph node is an example of secondary lymphoid organ. So you saw over here the WBC saw the pathogen for the first time. Here it becomes active. So lymph nodes are the spleen and everything. They come under secondary lymphoid organs, and these are the primary lymphoid organs, just bone marrow and thymus. Works? I hope it works. and I hope I have written the answer. Yes. So lymph nodes are small oval structures located in the lymphatic vessels. They act as filters trapping pathogens, foreign particles and cancer cells from the lymph. Lymph node contain lymphocytes, B and T lymphocytes, and macrophages. Macrophages are another type of WBCs. Okay, lymphocytes are another type, macrophages are another type. During infection, lymph nodes may swell due to activation of and multiplication of the immune cells. Works. Now thymus is a primary lymphoid organ located behind the sternum. Sternum, as I said, it's the breastbone on which the ribs are attached. Okay, it is responsible for maturation of T lymphocytes, which are crucial for immune response. The thymus is active in childhood and in early adolescence, and later its size, it slowly shrinks. Okay, so it shrinks after puberty, which is also given in your NCERT. I hope you write this and come. Okay, 3-marker question, write it properly in points and come. I hope it's clear, children. Going on to the next thing. Differentiate between humoral immune response and cell-mediated immune response. Okay, draw a schematic diagram of an antibody molecule and label any 4 parts. We already did it, I am not doing it again. Differentiate between humoral immune response and cell-mediated immune response, I will tell you. So, in the case of humoral, in the case of cell-mediated, humoral immune response is where your humor is there. Humor as in not your sense of humor, humor as in fluids. Your fluids are involved. What fluids? Basically your blood and everything is involved. So you know in our blood we have antibodies. So whenever we say that antibodies are being released to kill something that is there in our blood itself, you call this as humoral immune response. Cell mediated as in when cells are being involved. So antibodies are released by B cells. So over here, B cells are the major cells in action. They are the major cells which are doing the whole action. Over here, cell-mediated immune response, you know that cells comes together, they do the immune response in the case of T lymphocytes. In the case of T lymphocytes, you have seen them fighting, right? Either they release everything, perforins and everything, they kill the cells, or TH cells are there which activate other cells to be active, and then B cells becomes active. So this is not cell-mediated, where cell itself is doing everything. Over here, in the case of humoral immune response, the blood has antibodies and everything, it has the pathogens also, it kills everything there itself. Here it is mediated by B cells, here it is mediated by T cells. That is the difference. But you have more differences and I have made it over here. Okay. See, humoral immune response involves B cells and the production of antibodies, involves T cells, specifically cytotoxic T cells, the one that kills. Okay. Antibodies are released into the blood and the lymph, that is the humor. T cells directly attack the infected or abnormal cells, that is C cells, right? Now, primarily defense against extracellular pathogens. This is an extra point. If you want to write it, you can write it. Okay. Primarily defense against extracellular pathogens as in the cells that is coming from, that is outside the cell. That is outside the cell. So, bacteria and everything, this did not get inside our cell to affect us. From outside only it causes all the trouble. But in the case of virus, what does virus do? Virus gets inside our body, uses our mechanism, uses our cell's mechanism to do everything. So this is primarily for extracellular pathogens like bacteria and other things, viruses that are outside the cell. But over here it defends against intracellular pathogens, the ones that are inside the cell. They kill the cell from within. So virus inside the cells and cancer cells. So T cells and everything, they are very much active in killing cancer cells also. Okay. So that is one thing. Going ahead, memory B cells provide long-term immunity by quickly producing antibodies during future infection. This is for the memory B cells. Obviously here memory B cells will be produced, here memory T cell will be produced, which will provide immunity by rapidly responding to previously countered pathogens. It will just directly go and kill it. Okay. Effective against pathogens in body fluids, whereas effective against infected or abnormal cells directly. It's the same point, but yes, some other way. So you can use all these points to write in your answer. You can cut 2 points because it's a 3-marker question, and over here you are asked in the second part to draw a schematic diagram also of the antibodies, so you don't have to be making that there. Works? So you can write it for 2 marks. It's a 3-marker, so basically don't write too much in points. 2 points for this and make the diagram for this. Works? Going ahead. Okay. Next question. What are lymphoid organs? Write the functions of the following in response to immune system in our body. Bone marrow, thymus, spleen and lymph nodes. So lymph nodes we have studied, thymus also we have studied, bone marrow we'll study a bit and spleen also we'll study a bit. Okay, are you ready? So let's see what are the lymphoid organs. I already told you there are two types of lymphoid organs. One is primary lymphoid organ, the other is secondary lymphoid organs. So primary lymphoid organs are the place where all your cells are being made. Your B cells, your T cells, everything. Okay, and then secondary lymphoid organs are where they get activated. All your cells get activated. Okay. Now the example of primary, one is bone marrow. So if you have ever seen a bone, okay, just a second. Okay, this is a 5-marker question. Now we have entered the 5-marker question, so we have to be a bit elaborate over there. So this is a bone, suppose. Okay, inside the bone there is something that is known as red marrow. So if you are non-vegetarian, you might know it. Okay, inside the bone there is red marrow and there is also yellow marrow. Yellow marrow helps in making more bones and making more fat and stuff like that. Basically over here, red marrow has certain cells which are known as stem cells. What type of stem cells? Hematopoietic stem cells. Hematopoietic stem cells, that means the ones that makes the blood cells. Okay, what does this hematopoietic stem cells do actually? Basically they give rise to RBC, they give rise to WBC, they give rise to platelets, all the blood cells that you can imagine. Okay, stem cells, they have the potency to give out different type of cells. Now RBC is fine over here, they mature here itself. WBC is also fine, platelets is also fine. But in the case of WBC, there is one type of WBC, as I told you, lymphocytes. Okay, WBCs, no, there are different types. One is lymphocyte. Another, should I be telling about this now? Okay, listen to me. Lymphocytes are there. You have B and T lymphocytes. Apart from that, you have macrophages, you have neutrophils, you have eosinophils, you have basophils. Everyone have different functions. So for example, I have allergic response. At this point of time, my eosinophils get active. That is a cell that gets active. That is a type of WBC that gets active. Okay, when inflammation is required, basophils get active. Okay, when something is there inside our body and I want to kill it, take it inside, I have macrophages or I have neutrophils. So you have different cells for different functions. Okay, so one such type of cells is lymphocytes. In that you have B and T lymphocyte. B lymphocyte says that I will mature over here. T lymphocyte, as I said, goes to the thymus and gets matured there. Cool? Works? That is about bone marrow and thymus. Going ahead with secondary lymphoid organs, the ones that is given over here, it is spleen. To explain, you have spleen and you have one more thing you had. What was this? Spleen and lymph nodes. Lymph nodes I already explained you. Spleen, listen to me. So spleen is an organ which is there on the, for example, this is the stomach. On the top of this you have spleen. Spleen is told as graveyard of RBCs. It is known as graveyard of RBCs because this is where the old worn out RBCs come and die. So you know, after 120 days, the lifespan of RBC is just 120 days. After 120 days it comes to spleen. Okay, when it travels through, it's not like it is coming to spleen, but when it travels through spleen, no, what happens? There are certain changes in their feature. So there will be, in the spleen you have so many WBCs because this is where they get active. WBC sees them, kills them. The RBCs are killed. It uses all the important thing, it takes the important things and rest is gone. Okay, so that you have to make other RBCs. Similarly, spleen is a place, as I said, WBCs are there. So there will be pathogens also that comes along with the blood. These pathogens are also killed in the spleen itself. So even though it's known as graveyard of RBC, but still other functions are also taking place here, over here, because it has enough amount of WBCs. So wherever you have enough amount of WBCs and wherever they are acting for our protection, it is known as secondary lymphoid organs. Works? So over here, this is the answer that you have to give. Bone marrow is the primary site for production of blood cells, okay, including red blood cells, white blood cells and platelet. It also produces stem cells that can differentiate into various immune cells. So you just have to write until over here, okay. Now next thing, thymus, an organ where T lymphocytes mature. T cells plays a crucial role in adaptive immune response, basically same thing, that is you are talking about cell mediated you're talking about cell mediated, or you're talking about acquired immunity that you acquire throughout your lifetime by recognizing and respond to specific pathogens. Okay. Now spleen, what is an organ that filters blood, removing old and damaged red blood cells. It also helps in immune responses by storing white blood cells and acting as a site where immune cells can detect and respond to pathogen. Lymph nodes, again, it's a small bean shaped structure, okay, that filter lymph, I won't say it's a bean shaped structure, say it to be oval shaped, okay, structure that filter lymph fluid that circulates through the lymphatic system. They trap foreign particles such as pathogens and are a key site for immune cell activation. Just like that you have to explain. Then you have something that is known as Peyer's patch also. Peyer's patch. Okay, Peyer's patch is the mucus layer which is there in the small intestine. So all this mucus layer, wherever we have mucus layer, for example, respiratory tract we have mucus layer, all of them together we are known as mucosa associated lymphoid tissue. Okay, mucosa, you call it mucosa associated lymphoid tissue. So mucosa is the innermost layer of our body which has the mucous membrane and everything. So associated to it, if you have any tissues which helps in protection, you call them mucosa associated lymphoid tissue. Peyer's patch is one among them. You have tonsils, tonsils is one among them. Okay, Peyer's patch is in small intestine, tonsil is there in the neck region. Okay, they will also have enough amount of lymphocytes, as that something comes in, they pick it up, which is why we have tonsillitis. Okay, Peyer's patch is there in our small intestine, there also it picks things and kills it. Work, they are all coming under the secondary lymphoid organs. This also comes under secondary lymphoid organ. Okay, only this and this is the primary lymphoid organ. Going ahead, explain different techniques which helps in cancer detection and diagnosis. So what are the different techniques that helps in cancer detection? This is a 5-marker question, so you have to answer accordingly. I'll explain it to you. So you have these many techniques, my dear children. Okay, one is biopsy and histopathological study, second is computer tomography, third is radiography, fourth is MRI, and fifth is antibodies. So what do you mean by biopsy? I have explained it previously in some other things. Okay, so over here, suppose that person is having cancer. Okay, and there is a lump over here in the hand. Okay, so this lump is being taken, a part of it being cut. This part is used, right? This is known as biopsy. When I am doing this, when I am cutting it, I am taking it out, this is known as biopsy. Now, when I am taking this tissue, obviously this is a tissue, it will have number of cells, it will have number of cells, and now I am studying this tissue, and now I am studying this particular tissue. At this point in time, when I am studying this particular tissue, it is known as histopathological study. So instead of making the arrow down, I'll say this, because histo means what, children? Histo means tissue. Pathological study means you are studying you're studying it fully in the laboratory. So biopsy is done, okay, and then you are doing histopathological studies. You're seeing whether their cells are just growing around rapidly, immediately, okay, not stopping. This is what you mean. Okay, you're checking for the cell's behavior. Fine, this is one way to detect, and you know what are the features of cancer cells that could be seen very evidently. If you do not know, do not worry, I have a question for that also. We will be talking about that. The next one is computer tomography, or you call it radiography. Okay, this is the next thing. So you do not call it radiography, I'm so sorry. Okay, computer tomography is there, and then you have radiography. Computer tomography as in CT scan. Okay, and radiography as in when you are taking X-rays and everything. Over here you have to understand one thing, that in both of this thing, you are using ionizing radiations. That is X-rays itself. You are using ionizing radiations. Okay, one will be giving you 2D image, other one won't be giving you 2D image, so you won't be able to find it out. Okay, so computer tomography is there, radiography is another mechanism that you are having, but you are using ionizing radiation, which is not very good for your body again. The next one we have is MRI. MRI stands for magnetic resonance imaging. Okay, where basically you are using magnetic field. You are using magnetic field to analyze the disease, and this is considered to be one of the safest procedure. Why? Of detecting things. Why? Because over here you are not using ionizing radiations, rather than that you are using non-ionizing radiation. You are using what? Non-ionizing radiations. Fine. Now going ahead, you have antibodies. That is next way. So I told you, every time something gets inside our body will be releasing antibodies. Our body will be releasing antibodies specifically for that particular antigen. So I know what are the antibodies that will be released for cancer. So those antibodies, I have it already. I have those antibodies already. Okay, specific antibodies. Now what do? Basically, if the person who is there, who is suspect of having a cancer, that person's cells are given and I am checking for antigen, and if that antigen is specifically binding to this antibody, that means what? This particular person is having cancer. If the antigen is not binding to the antibody, there is no cancer. If it is binding, then yes, that particular person has cancer. So these are the 5 different ways which will help to detect whether a person has cancer or a person does not have cancer. Works? Okay, I hope you will be able to write it as well. Going ahead, the next question, 23rd question, and then we have 3 more children over here. Name and explain the property present in normal cells but lost in cancer cells. Okay, so we have to talk about the properties of cancer cells. All normal human cells have genes that may become cancerous. So let's take one by one. So name and explain the property present in normal cells but lost in cancer cells. Let's see the properties. So over here you have these many properties. This is a normal cell, this is a cancer cell. First property is contact inhibition. That means when a normal cell is there, when normal cells they divide, okay, suppose from here division is starting, there, okay, just a second, from here also division is starting for some other cells. They divide. When they reach over here at this particular point of time, they stop the division because they are coming in contact, and this is known as contact inhibition. What do you call this? You call this as contact inhibition. On the other hand, when you talk about cancer cells, okay, what happens? Suppose this is a tray again. You have normal cells over here. They are dividing, okay, and then you have cancer cells over here. They are also dividing. Fine. What will happen is that the cancer cells, they keep on dividing and they would not show something that is known as contact inhibition. They have lost the property of contact inhibition. So I am writing no contact inhibition that is being seen over here. Okay, so that is first property. Second is metastasis. What do you mean by that? Cancer cells basically says that we cannot stay at one place. Cancer cells, they are very much unique in their own ways, and they say that we cannot stay at one place. We will move from this, for example, this is hand region and this is stomach. So from here to here it will be moving, and this particular property is known as metastasis. You have to remember one more thing, cancer is of 2 types. One is benign. Okay, you can say tumor is of 2 types. One is benign and the other one is what? The carcinogenic one. Oh, sorry. Malignant one. Okay, so one is benign and the other one is malignant one. The benign one just stay there. It forms tumor. So cells basically when they divide, these cancer cells when they divide, they form a structure like this, a mass of cells which you call as tumor. Now if the cells of the tumor are not moving anywhere, they are not showing metastasis, I call this as benign tumor. And the cells of the tumor, if they are moving to different places, I call this as malignant form of tumor, and this is children cancerous. And this is cancerous. They are moving around somewhere, everywhere. So metastasis is a property that is shown by cancer cells. Loss of contact inhibition, that is also shown by, please don't write contact inhibition is shown by, please write loss of contact inhibition, okay? Loss of contact inhibition is shown by them, and metastasis is also shown by them. Now in them they have something that is known as proto-oncogenes. Okay, in normal cells we have proto-oncogenes. What do you mean by proto-oncogenes? Basically when they are there, okay, they normally help this okay, so basically we have DNA, you have DNA. This DNA has specific regions, you call them as genes, and these genes basically code for proteins. These genes basically they are going to code for proteins, right? So over here over here in this case, what is happening is that there are certain genes which are known as proto-oncogenes. So proto-oncogenes, when they are normally working, they give rise to different proteins which are actually good for the cell. They are all fine. But in the case of cancer cell, what happens is that this particular proto-oncogene that you see, it will have certain mutations. This particular proto-oncogenes that you see over here. So over here you had the proto-oncogene and it was acting all cool. But here you have some mutation in the proto-oncogenes such that now they will be acting like, they will be causing cancer. They will be making unwanted proteins. They will be making unwanted proteins that will lead to rapid cell division, that will lead to rapid cell division and things like that. So as of now they were working fine, but when they got mutated, okay, when they got mutated, they are not working fine. They are making the cell do cell division and things like that repeatedly. So that is proto-oncogenes. So they will be having activated proto-oncogenes or mutated proto-oncogenes. You can write, they can have mutated proto-oncogenes, but it's not a property. It's not a property. So you can write contact inhibition and metastasis as a property. There is one more property, telomerase activity. I don't think that is needed for you in 12th grade. So as of now, I will not explain it to you. Okay, if you want me to explain, you can tell me, I can explain in the comment section. Okay, otherwise the time will be just expanding. So for children, you guys, you just have to write both of them. Okay, this is a cause of cancer. You can say that this proto-oncogene is a cause of cancer. When they get mutated, it can cause cancer. These are the properties that you need to remember. Loss of contact inhibition and metastasis. Works? Next thing, all normal human cells have genes that may become cancerous under certain condition. Name and them and explain how. So I just explained that now. So that is about proto-oncogenes. Proto-onco. Onco as in the one that cause cancer, right? Proto-oncogenes. So usually they help in cell growth. They help in formation of proteins and lot of things. But once they are getting mutated, they make the cell turn into a cancerous cell. So some mutation in them can lead to the whole change. Okay. Now state the role of following techniques in detection and diagnosis of cancer: biopsy and histopathological study. I told you already. And magnetic resonance imaging. They are using the magnetic field to understand things. In biopsy, you are cutting the part and you are examining it. This process is known as histopathology. Okay. Done. 23rd question done. I hope you will be able to frame answers. So framing answers, obviously in every question I won't be telling you how to frame answers. You should be knowing it. But if you have to, the first thing that you have to do is understand the concept. Without understanding the concept, you cannot frame the answers. So please understand the concept, and I hope I am helping with that thing at least over here. Yeah, works? Now going ahead, children. Okay, there's a long type question, 24th one. Read the following passage and answer the questions that follow. Okay, in 1981, the health workers of United States of America had become aware of sarcoma. Sarcoma is cancer, okay? Cancer of the skin and blood vessels, and another disease, pneumocystis pneumonia, a respiratory infection caused by a protozoan. Okay. Both these diseases were very rare in the general population but occurred frequently in more severely immunosuppressed individuals. So it is said that it is very rare. Both these diseases that you see, that I told you right now, it is very rare to be seen, but it is rare as in you won't find it often, okay? But it was said that few people who were immunodepressed, as in they were not showing too much amount of immune response, their body was immunologically weak, those people were getting this disease, okay? This led to recognition of immune system disorder that was named acquired immune deficiency syndrome, that is AIDS, okay? Then it was discovered that people who are having this, they have low immune response, or their body is not immunologically active, which is why they are getting any random disease, okay? Now, in 1983, virologist working in the USA and France has identified a causative agent for virus HIV, the disease. Name the group of cells that the HIV attacks after gaining entry into the human body, and write the various events that occur within this cell. So that is thing. So you have to name the cells, and you also have to explain what are the events. So for that, I have come across with this. Chalo, let's study. So this thing that you see over here is HIV virus. Okay, HIV virus will be having a membrane around it, an envelope around it. Okay, an envelope around it. This envelope is made up of our membrane lipids. This is made up of our lipids, such that such that something that gets inside our body would not be identified because it is using everything of ours. Okay, I'll tell about that. Now over it you see things like this which looks like lollipop. This is known as glycoprotein 120. This is a protein. This is a protein, you call it GP120. And inside this you see one very tough covering which is known as capsid. So all the viruses usually they have a thing that is known as capsid. And inside the capsid you will be finding the genetic material which is over there. It's an RNA. They have what, children? They have RNA and they have two single stranded RNA. I'm not talking the single stranded. There is one strand, there is another strand. They are not complementary to each other, okay? They just have two single stranded RNA. And apart from that, you see the colorful stuffs over here. Basically, they have enzymes. Basically, they will be having enzymes, children. How many of them? 3 of them. I call it RIP because they kill our cells. I call it RIP. You'll get to know what is RIP. Suppose now the HIV virus is there. It has come. It has come inside our body, and now there are certain proteins on our cells. Okay, there are certain proteins on our cells onto which, I'm not writing the name of the proteins, basically onto which these guys comes and attach. This virus, it comes and attaches with the help of the GP120 protein. With the help of the GP120 protein, it comes and attaches on this particular thing. I call this cell as macrophage cell. Macrophage cell. Macrophage cell, what does it do? It takes in everything. Similarly over here, it is taking in the HIV virus. So let me take it in. So over here, what happens is that basically the HIV virus, it sends out everything inside. So what do you have inside now? You have the RNA and you have the enzymes. You have the RNA and you have the enzymes over here. Works? So, and this is a macrophage, this is a normal cell. So it will be having its own nucleus and its own DNA as well. It will be having its own nucleus and own DNA as well, right? So this RNA is there. This is RNA is belonging to the HIV. HIV things, the human immunodeficiency virus. Okay, that is HIV's full form. Human immunodeficiency. It is making the human immunodeficient. This virus thinks, I want, anyway I got inside, now I want to be just like them. I want to use their machinery. So what do? But I have RNA and they have DNA. So shall I just transform my RNA into DNA? Can I make a DNA out of it? So the thing is, yes, they can make a DNA out of it because they have the first enzyme which is known as reverse transcriptase. Reverse transcriptase. You know transcription, what do you mean by transcription? Basically, when DNA helps in the formation of RNA, this is known as transcription. This is known as transcription. What is this? This is a DNA. Now, ulta of this is happening. RNA is now going to make a DNA. RNA is going to make a DNA. This is known as reverse transcription. This is done by reverse transcriptase. So, I am making a DNA strand over here. Okay, fine. Now, this DNA again will be having a complementary strand. You don't ask me how. I'll tell you later on if you want. Basically, there is the reverse transcriptase enzyme only helps in the formation of another complementary strand, such that now the DNA, it looks like this. Same. The DNA is just like that. Okay. So, virus's first function is done. First function was to use the reverse transcriptase to do what? To make DNA from RNA. That is done. Now, this DNA has to get incorporated there. So, I am just changing the color a bit so that you understand things better. So, this DNA, I am changing it in green color. Okay. Now, this guy has to get accumulated there. So, it gets inside and now it uses a second enzyme that is over here, which is known as integrase. So, RIP, R stands for reverse transcriptase, I stands for integrase. Okay. So, RIP goes there, cleaves the DNA with the help of integrase and gets integrated over here. Now, the nucleus will use its machinery to form copies, to do transcription and to make the proteins and everything. So, all this thing happens, and now if you see, proteins are made Okay, the mRNA is basically made for both this strand and also the viral DNA strand. So viral DNA strand also make the mRNA moves outside. Okay, the mRNA, this is viral mRNA. And if you remember, after DNA makes RNA, this RNA is used to make what, children? Proteins. So now by using this mechanism, I made the mRNA, and now mRNA basically uses ribosomes and everything and makes what? Proteins. What proteins am I making? The viral proteins only I am making. I am making capsid and everything here itself. Whatever proteins are needed, that everything I have been making. Now these proteins to be active, no, you need certain enzymes to cut them off into smaller fragments. Okay, so you call them proteases. What do you call them? Proteases. So that is the third enzyme that we are having. Proteases. So over here, the proteases basically cuts these proteins into smaller fragments. Cool? Works? Okay, and now they are all active. So you have this protein enzymes that you see over here, they're all proteins only. Enzymes that you see over here, they're all proteins only. So basically the virus is using all the machinery to make the proteins itself. So you have all the proteins now. And from here also you get the RNA also. Okay, you make the capsid protein also, and slowly this guy will be moving out in the form of a virus. Again, it will go and affect the next cell. This goes on, goes on, goes on. So the first cell that it affects is macrophage. The first cell that it is affecting is macrophage. Now, the next cell that it will be affecting will be T helper cell. Please remember, it's very much important. So first cell macrophage, second cell is T helper cells, and later on they will kill all the T helper cells, which is why our immune response becomes less if one person has HIV infection. Okay, in HIV infection, they are targeting the cells which protects us, and that is T helper cell. You saw how T helper cell was working around. So if this is affected, obviously nothing will be working around. So T helper cell and macrophage will be affected. First this and second this. This is the reason. This is the reason why the immune response will be very less. I hope you will be able to explain this in the form of an answer. Okay, done. Now, name the group of cells. The HIV attacks children, it is macrophage and TH cells, all the immune cells basically. Okay, and then write the various events that occur. So basically the virus gets inside the macrophage and later uses reverse transcriptase to form the DNA. This DNA goes and integrates into the DNA of the normal nucleus and then forms the mRNA. This mRNA forms the protein. Okay, and you have to tell that proteins are made, and then later on this HIV virus moves out to the cell and goes affect the next cell. Simple. Okay, can you frame it? I have not written it, but I have told you. Now, write the expanded form of the diagnostic test used for detecting AIDS. I already told you it's ELISA. So it will be, enzyme linked immunosorbent assay. ELISA. Cool. Now, write the possible treatment available for disease at present. So, do you have any big treatments available? Can you cure AIDS? No, you cannot cure AIDS, but you have antiretroviral drugs. Okay, which helps in not causing too many diseases. So they will be in a stable condition because of this disease. Okay, this particular drugs. So antiretroviral drugs are there which are used. Fine, that is a possible treatment. Now mention any 2 steps suggested by WHO for preventing the spread of this disease. One is blood bank is made safe. Okay, such that anyone who have, for example, people who have thalassemia and anything where you have blood transfusions to be made every now and then, if the blood itself is contaminated, the other person will also have it. So blood banks are now safe. The second thing, disposable syringes are being used now. Disposable syringes are used so that none of the people, they repeat the syringes, and that's how the blood will get mixed and you could have disease. So that could be avoided. Done? Done with that? The next portion. Over here, 25th question, the last question that we are having. I hope you are not tired. I get you. Even I am speaking for a long time and my tongue is getting twisted. But yes, I think you need it very well because your exams are coming near and you need the answers and the questions as well. So here I am. So describe the life cycle of Plasmodium from the time it enters the human body till a female Anopheles mosquito bites an infected person. Mention the 2 events of Plasmodium life cycle that occur within the female Anopheles body. So we have to talk about the life cycle, and then we have to talk about the 2 events that is occurring in the female Anopheles body. So easy thing is this. I made it initially. Listen, Plasmodium is the one that causes malaria, and this Plasmodium that you see is a protozoan. It's what? A protozoan. It's a eukaryote. It's a eukaryote. It has nucleus and everything. Okay. So what it does is that this Plasmodium gets into Anopheles mosquito, and in Anopheles mosquito, it is in this particular stage. You call this sporozoite stage. What do you call this stage as? Sporozoite stage. Okay. In this stage, it is there in the salivary gland. So mosquito is there. It has saliva, right? And in the saliva, what do you have? Sporozoite stage of the Plasmodium. Will you remember it? You have to remember it. Okay. Now, the sporozoite, which is there in the saliva. Now, the mosquito is there. I am the mosquito, and I am going to bite a human being. Not like this. Okay. So, basically, when I bite, I give certain saliva there. I don't give blood, obviously, because I want blood. I give the saliva such that my saliva has anti-clotting enzymes so that I get enough amount of blood. Okay. So, when I give the saliva, basically, I am giving the sporozoites also. So, the sporozoites which are there are getting inside the human body. Where in the human body? In the blood. It is going in the blood, right? I have given, now it's there in the blood. From this area, from the blood, it goes to the first camp. So think of it as a terrorist. So this sporocyte is going to the first camp, that is liver. Okay. In the liver, you know, liver cells, the name of the liver cells are known as hepatocytes. What do you call it? Hepatocytes. In the hepatocyte, they will think that we have to increase our number, yeah, this won't work. So they do asexual reproduction. Obviously, when number becomes more, the hepatocytes will break open. The hepatocytes, they will break open, right? Hepatocytes, when they break open, all this sporozoites or whatever stage it is, we call it sporozoite only for your understanding, they just get spread into the blood. So blood becomes their second camp. I'm telling you again, Plasmodium is there. Okay, Plasmodium is there in the Anopheles mosquito's salivary gland in the form of sporozoite. The sporozoite is given to the human being, okay, while the mosquito came and bit it. Okay, this particular sporozoite is going to the liver, that is the first camp. In the liver, the hepatocytes or the liver cells are affected. They undergo asexual reproduction there. The hepatocytes, they get lyased, okay, and then what will you be having, children? Basically, all the cells will be going to the blood. In the blood, they will be going to the RBC, red blood cells. In the blood, you have so many cells, but they are specifically going to the RBC. Again, they are doing like, we have to divide our cells again. So they do asexual reproduction again. Now the RBC also will not be able to tackle all of them, obviously. So it will get lyased. When it gets lyased, children, hemozoin is being released. Hemozoin is being released. Actually, I'll tell you what happens. This particular Plasmodium, no, basically they are feeding on the hemoglobin. In the RBC, they are feeding on the hemoglobin, and when they break hemoglobin, they get heme and globin. But this heme, it's actually very, in this this heme, when they break, they convert into hemozoin, which is very, basically heme and globin is there, but this heme, if it stays like that, it will be very toxic. So they convert into something that is known as hemozoin, which is less toxic. But then when it was doing all this drama, at this point of time, it got lyased, and hemozoin is also now released. Hemozoin is very toxic to us. So what will happen? Basically, we will have fever and we will have chills. This is the reason that in malaria, we get chills, because of what? The release of hemozoin. So while the Plasmodium was having the RBC, was feeding on our hemoglobin, at this point of time, hemozoin is made with the help of hemoglobin itself. I told you, hemoglobin breaks into heme and globin. Heme is toxic, so it converts into hemozoin. It's just keeping there. But then since it is getting lyased, hemozoin gets released, and now we have fever and chills. The second instance is that basically while doing asexual reproduction and everything, they are getting converted into a different stage, which is known as gametocyte stage. Okay, they are forming gametocytes. They are not gametes. They are not gametes. This is a stage that is known as gametocyte stage. Now, these gametocytes basically are the ones that will be forming the gametes later on. As of now, you see they are maturing, and now they have reached the stage of gametocytes. They are adults now. You can think like that. Okay, these gametocytes are still there. They say, only when a mosquito comes and bites me, inside the mosquito, I will form the gametes. So mosquito comes and bites, takes the blood. In the blood, what do we have? We have gametocytes. The mosquito's blood goes to the gut, mosquito's gut, not mosquito's blood. Our blood goes to the mosquito's gut, right? In the mosquito's gut, what will happen, children? The gametocytes which were there, it will form the gametes. It will form what? The gametes. Now gametes are formed in the gut of the mosquito. Okay, but where do you find sporozoites? In the salivary gland. So something might have happened in the gut. So in the gut, what happens basically, the gametes, they fuse to form the zygote. The gametes, they fuse to form zygote. Okay, and the zygote will now form something, convert into something motile. Motile as in something that can move, and it will slowly move up to the salivary gland. So the two events that is happening in the case of mosquito is that basically the blood that is got from the human, it has gametocytes. So in the case of mosquito, in the gut, the gametes which are being formed with the help of gametocytes, they are fusing together, they are forming the zygote in the gut. Okay, second thing that happens is that this zygote, some of them, the motile form of it, they will be moving to what? To the salivary gland in the form of sporozoite stage. So zygote itself is not moving, it develops into something else, and that something else later forms the sporozoite which is moving to the salivary gland. Works? I'll explain once more, listen to me very carefully. So I am the mosquito, I have the sporozoites, okay, in my salivary gland. I gave the sporozoites. Now I am the sporozoite. I went to the liver first. In the liver, I damaged all the hepatocytes. Okay, what did I do to damage? Basically, inside the hepatocyte, I divided myself. I did asexual reproduction. So I have so many of us like me, okay. And now the hepatocyte got sliced, and I now move out. I go to the next cell, that is the red blood cells. Okay, in the red blood cells, again I do asexual reproduction. Again, the red blood cell also gets sliced, but this time I was having food also. And what was my food? Hemoglobin. And this heme I was converting into hemozoin because this heme was toxic for me. But during this process, when the cell got lysed, hemozoin got released, and this hemozoin was the one that causes fevers and chills in the body. Okay, the second thing that happens is that I said that I am undergoing asexual reproduction in the RBC. So I am making a different type of me that is known as gametocyte. It's a different stage. Gametocyte is there in the blood. Okay, now this gametocyte basically is taken by the mosquito again. This mosquito drinks it, I am the mosquito. Now inside my gut, okay, inside my gut, what is happening, children? These gametocytes are going to form gametes. They finally form gametes. The gametes, they fuse, they form zygote. Now from the gut, from my gut, this particular zygote develops into something else and later comes in sporozoite stage to my salivary glands, and again I go bite another human being, and the same cycle continues again and again and again. So two events is that in the mosquito's gut, the zygote is formed when the gametes fuse. Second event is that sporozoites are being transported to the salivary glands in the mosquito. Rest everything is same. So describe the life cycle of Plasmodium. I hope you got it. And with this, children, we are ending the whole thing, and I hope that this particular video was really helpful for you. I tried my level best to explain everything in as much as detail as possible, and you have a quick revision of all the things which is there in the chapter. If you think it was useful, definitely do let me know, and if you think that there are things that you still need to understand, you can put it down in the comment section. I will be really happy to help you out as well, and I wish you all the best for your examination, and do not forget to subscribe to our channel as well.